It is June the 26th, 2025, and my name is Jasmine Gillette. Welcome to the final project on Chapter 37 from Harper's Illustrated Biochemistry. Today we'll explore the intricate process of protein synthesis and the genetic code. This chapter is foundational to understanding how genetic information is stored in DNA and is ultimately used to produce the proteins essential for life. Over the next 20 slides, I'll take you through the flow of genetic information, the mechanics of translation, and key biomedical insights such as the effect of antibiotics and genetic mutations. These are our learning objectives. By the end of the presentation, you will be able to explain the central dogma of molecular biology and its significance, describe the structure and components of the genetic code, interpret the codon table and explain codon degeneracy, outline the rules of tRNA, amino acyl tRNA synthetases and translation, analyze the steps of protein synthesis, initiation, elongation, and termination, explain how ribosomes function as translation machinery, discuss key regulatory mechanisms in protein synthesis, identify important post-translational modifications and their functions, evaluate how mutations affect protein synthesis, and understand the mechanisms by which antibiotics inhibit translation. The central dogma of molecular biology is the concept that genetic information flows in a single direction, from DNA to RNA to protein. In the nucleus, genes are transcribed into messenger RNA, which then exits into the cytoplasm where it is translated into a specific protein. This process ensures that the instructions encoded in DNA result in the synthesis of proteins that perform vital cellular functions. DNA remains the stable repository of genetic instructions, while RNA acts as the messenger conveying this blueprint to the ribosomes. This flow is essential for life because proteins serve structural, enzymatic, signaling, and regulatory roles. Any disruption in transcription or translation can result in abnormal protein synthesis, which may cause disease. So understanding this flow also forms the basis of many biotechnological and medical advances such as gene therapy and targeted drug design. The genetic code is composed of codons, which are 3 nucleotide sequences in an mRNA that specify amino acids. With 4 RNA nucleotides, there are 64 possible codons. Of these, 61 codons code for the 20 standard amino acids, while 3 function as stop signals to terminate translation. The start codon AUG codes for methionine and signals the beginning of protein synthesis. The genetic code has 4 critical properties. It is degenerate, multiple codons for 1 amino acid, unambiguous, each codon specifies only 1 amino acid, and non-overlapping, each base belongs to 1 codon. Finally, it is nearly universal, used by almost all organisms. These characteristics make the genetic code both robust and efficient and minimize the impact of mutations while ensuring consistency across life forms. This is the codon table and degeneracy broken down. tRNA and amino acyl tRNA synthetases. Transfer RNAs act as molecular interpreters of the genetic code. Each tRNA has an anticodon region that base pairs with a complementary mRNA codon and a 3N where a specific amino acid is attached. The attachment is facilitated by enzymes known as amino acyl tRNA synthetases, which recognize both the correct amino acid and its corresponding tRNA. There are at least 20 amino acyl tRNA synthetases, 1 for each amino acid. These enzymes are highly specific, ensuring that the correct amino acid is loaded onto its corresponding tRNA. Mischarging is rare, but it can be detrimental. This precise pairing of tRNA and amino acids is crucial for accurate protein synthesis. The Wobble hypothesis explains why fewer tRNAs are needed than the number of codons. The third nucleotide of the codon, known as the Wobble position, is less stringent in its base pairing rules. For example, inosine in the tRNA anticodon can pair with U, C, or A in the mRNA codon. This flexibility allows a single tRNA to recognize multiple codons that code for the same amino acid. It contributes to the degeneracy of the genetic code and enhances the efficiency of translation. Despite this leniency, the first two bases of the codon-anticodon interaction remain critical for specificity. Ribosome translation machinery. The ribosome is a complex molecular machine composed of ribosomal and RNA and proteins, consisting of two subunits, the small 40S and the large 60S in eukaryotes. It facilitates the protein synthesis by decoding mRNA codons and catalyzing peptide bond formation between amino acids using its A amino acyl, P peptidyl, and E exit sites to guide tRNA movement during translation. The initiation of translation is the first phase of protein synthesis in eukaryotes, where the small ribosomal subunit 40S assembles with the initiator tRNA, met-tRNA, and various initiation factors to form the 43S pre-initiation complex. This complex then binds to the 5' of the mRNA, facilitated by the EIF4F complex, and scans the mRNA in the 5' to 3' direction to locate the start codon, AUG. Once the AUG codon is recognized, the large ribosomal subunit 60S joins to form the 80S initiation complex, positioning the initiator tRNA in the P site and marking the start of elongation. The elongation of the peptide change. During elongation, amino acids are sequentially added to the growing polypeptide change. An amino acyl tRNA matching the next codon binds the A site, aided by the elongation factor EF1A and GTP. If the match is correct, GTP is hydrolyzed and the tRNA locks into place. Next, the peptidyl transferase activity of the 28S rRNA and the 60S subunit catalyzes a peptide bond between the amino acid in the A site and the growing chain in the P site. EF2 then promotes translocation. The ribosome shifts, moving the new peptidyl tRNA into the P site and the empty tRNA to the E site for release. Termination begins when a stop codon, UAA, UAG, or UGA enters the A site. Since there is no corresponding tRNA, release factors RF1 and RF3 recognize the stop codon and catalyze the hydrolysis of the bond between the peptide and the tRNA. This releases the newly synthesized polypeptide from the ribosome. The 80S complex then dissociates into its 40S and 60S subunits, ready to participate in another round of translation. This step is highly regulated to ensure proteins are completed before release. Polysome and translation efficiency. Multiple ribosomes can simultaneously translate a single mRNA, forming a structure called a polysome. This increases the efficiency of protein synthesis, allowing rapid production of many copies of a protein from one mRNA. Ribosomes are spaced around 35 nucleotides apart, minimizing collisions and maximizing throughput. Polysomes are commonly observed in cells with high protein demands, such as those producing antibodies or enzymes. Their presence is a sign of active translation. The regulation of translation MTOR pathway. The MTOR pathway plays a central role in translation regulation. MTOR promotes translation initiation by phosphorylating 4E-BPS, releasing EIF to 4E to form the EIF4F complex. This enhances ribosome recruitment to the mRNA cap. Under stress conditions like viral infection or amino acid deprivation, EIF2A is phosphorylated, inhibiting the formation of the 43S complex. This conserves cellular resources and prevents the production of potentially harmful proteins. These regulatory mechanisms allow cells to adapt to environmental changes. After synthesis, many proteins undergo post-translational modifications to become fully functional. These include cleavage, phosphorylation, acetylation, and glycosylation. PTMs influence protein folding, stability, activity, and localization. For instance, phosphorylation can activate enzymes, while glycosylation affects protein trafficking. Defects in PTMs are linked to diseases such as cancer and diabetes. Mutations in DNA can impact protein synthesis at multiple levels. A silent mutation changes a codon without altering the amino acid. Missense mutations substitute one amino acid for another, potentially altering function. Nonsense mutations create premature stop codons. Frameshift mutations caused by insertion or deletions alter the reading frame and usually result in non-functional proteins. Diseases like sickle cell anemia and cystic fibrosis arise from specific missense or nonsense mutations, highlighting the importance of accurate translation. Several antibiotics selectively inhibit bacterial translation. Tetracyclines prevent tRNA binding to the A site. Chlorophenol inhibits peptidyl transferase. Erythromycin blocks translocation. Puromycin mimics tRNA and causes early termination. These drugs exploit structural differences between prokaryotic and eukaryotic ribosomes, making them effective treatments with minimal toxicity. However, their use must be carefully managed to avoid resistance. In summary, protein synthesis involves decoding genetic information via a highly regulated process of translation. It begins with initiation, proceeds through elongation, and ends in termination. The genetic code structure ensures high fidelity in protein production. tRNAs and ribosomes coordinate to translate mRNA into the functional proteins, and regulation through signaling pathways and storage mechanisms allows cells to adapt to translation to their needs. Mutations in antibiotics highlight the clinical relevance of this essential process. These are some multiple choice questions. Each question tests an understanding of a key concept, from the role of codons and translation factors to the impact of antibiotics. These questions reinforce the core content discussed in this presentation. We'll go over this pretty quickly. For the answer to question 1 is AUG, because it is the initiation codon in eukaryotes, coding for methionine, and marks the start site for translation. The answer to question 2 is C, aminoacyl tRNA synthetase, because it is the enzyme responsible for attaching the correct amino acid to its corresponding tRNA and is a critical step in ensuring the fidelity of protein synthesis. Translocation, A, is the correct answer because EF2 facilitates translocation during translation and moving the ribosome along the mRNA to the next codon after peptide bond formation. The correct answer for 4 is C, nonsense. A nonsense mutation introduces a premature stop codon in the mRNA sequence, leading to early termination of protein synthesis and often resulting in a truncated non-functional protein. The answer to question 5 is B, chlorophenol. This inhibits peptide bond formation by binding to the 23S rRNA of the 50S ribosomal subunit in bacteria, thereby blocking the activity of peptidyl transferase and the enzyme responsible for forming peptide bonds during translation. These are the references. And then again, key takeaways, the genetic code, translation, regulation, clinical relevance. Thank you for watching this presentation on protein synthesis and the genetic code. I hope you now have a clearer understanding of how genetic information is transformed into the proteins that drive cellular function. If you have any questions or feedback, feel free to let me know in the comments. Thank you so much.